Ever wondered how cells regulate their cycle? Well, the answer lies in a fascinating process known as cell cycle regulation. This vital mechanism ensures that cells divide and reproduce in a controlled, orderly manner. At the heart of this process are enzymes known as cyclin-dependent kinases, or CDKs for short. These enzymes play a critical role in managing different phases of the cell cycle. But here's the catch. CDKs can't do it alone. They rely on cyclin molecules for activation. Without the binding of a cyclin molecule, a CDK is like a car without fuel. It's present, but it can't do much. Once a cyclin molecule binds to a CDK, it kickstarts the enzyme, allowing it to regulate various processes within the cell cycle. Intriguing, isn't it? This is just the tip of the iceberg. Let's delve deeper into how CDKs work. Now, let's talk about the mechanism of CDKs. These fascinating enzymes spring into action when they bind with cyclin molecules. This process is not a random dance, though. Each CDK has a specific preference for different cyclin molecules. Just like how we have our favorite dance partner, so do these CDKs. This preference is not without consequence. It's the binding of cyclin to the CDK that determines the enzyme's specificity towards its substrate. It's like the cyclin molecule whispers into the CDK's ear, guiding it towards its unique target. Now, the plot thickens. The mechanism of action of these enzymes might differ among different kinases. Each kinase regulates a different phase of the cell cycle, and just like an orchestra conductor, each kinase has its own unique approach to keep the rhythm of the cell cycle in sync. But what happens when things don't go as planned? When the dance of the CDKs and cyclin molecules is interrupted? That's when the cell cycle regulation comes into play, ensuring that the rhythm is maintained and the dance continues smoothly. So, that's how CDKs work. But what happens when they undergo changes? What happens when CDKs undergo changes? Let's find out. Imagine CDKs as keys and the interface as a door. When the keys undergo a change known as phosphorylation, they can turn the lock and open the door. This door leads to the inactivation of the retinoblastoma protein, or RB for short. But what does this mean for the cell? RB is like the gatekeeper of a castle, guarding the genes that encode proteins necessary for DNA synthesis. When RB is inactivated, it's like the castle gate is flung wide open. Multiple genes are then free to produce proteins that play crucial roles in DNA synthesis. However, it's not all a free-for-all. In this intricate dance of cellular processes, there are also CDK inhibitors, the regulators of the cell cycle. They function as the brake system of a car, slowing down or stopping the cell cycle when necessary. They achieve this by binding to the CDKs, preventing them from activating and thus negatively regulating the cell cycle. In essence, the changes in CDKs, whether it's the phosphorylation during interphase or the interaction with inhibitors, can greatly impact the cell cycle. It's a delicate balance, a dance of molecules that keeps our cells functioning properly. Changes in CDKs can have a significant impact on the cell cycle. But how is this process regulated? Finally, let's talk about how the activity of CDKs is regulated. The regulation of CDKs is a fascinating process involving the binding of proteins from the CYP-KIP families of inhibitors. These inhibitors play a crucial role in the negative regulation of CDKs by binding to these enzymes and distorting their cyclin interface and ATP binding pocket. This distortion prevents the enzymes from being activated. Now, you might be wondering why this is important. The answer lies in the cell cycle. When the activation of CDKs is prevented, it leads to a negative regulation of the cell cycle. This means that the cell cycle is slowed down or even stopped. This is crucial for maintaining the balance of cell growth and division and preventing uncontrolled cell proliferation, which can lead to diseases like cancer. So that's how cells regulate their cycle using CDKs. A fascinating process, isn't it?